Hello everyone, very good morning to all of you and welcome back to SRIRAM's IAS YouTube platform. So friends, we are trying to discuss about the concept of the one PYQ one day series. So welcome back to Anthropology PYQ series. So let's try to decode every question and let's master every concept. So friends, for us, the question with us today is, what are genetic markers, and discuss their applications in understanding population variation, disease association, and forensics. So this is the question. So the question is completely about the genetic markers. So it is one another important topic in our anthropology paper 1. So let's try to discuss about this particular question and let's try to know about this concept in detail, right? So first, let's try to start our discussion with how to write an approach for this question. So what we can write in the approach part of the question. So first, so what you can do in the introduction portion of the answer, you can simply define what are genetic markers with systematic examples. So first we can explain or mention what are genetic markers. Then coming to the body portion of the answer, so there the question is directly asking about the aspect of discuss with respect to the applications of genetic markers. So the question is very straightforward and you can just have it and you can just check it clearly. So applications of genetic markers in understanding population variation, applications of genetic markers in disease association, and applications of genetic markers in forensics. So this is the basic demand. And same thing you have just mentioned in the body portion also. So that's one thing. And then how you can just conclude in the conclude the question or answer. So you can simply highlight the scope, further scope and relevance of the genetic markers, and thus you can conclude the answer. So this is how you can keep your approach. So all these aspects you can mention in the introduction, conclusion, and the body portion of the answer. I hope that's clear with you. So did anyone try to just attempt this particular question? So if anyone has attempted, you can just comment in the chat box. So let's try to understand this particular concept in a systematic fashion. So first, let's try to understand what are genetic markers and some of the examples of genetic markers in the first instance. So there, first, if you happen to understand what are genetic markers. So friends, look here carefully and try to understand the concept carefully. Now, let's start from the basic understanding of the structure of a cell. So if you take up a cell, if you consider this as an animal cell, within an animal cell, at the center of the cell, there would be nucleus. So nucleus is nothing but the central part or central part of the cell. So within the nucleus, if you happen to see, if this is the nucleus, so within the nucleus, you can see some deeply stayed thread-like structures. So the deeply stayed thread-like structures which are present within the nucleus of the cell are known as chromosomes. Try to understand this. And within the chromosomes, we can see some part of some genetic information present in the form of small genes. So these genes are made up of different DNA. So now, if you happen to just extract the chromosome out of it, and from this chromosome, if you happen to extract the DNA, so DNA looks like this. So DNA is nothing but the hereditary material in certain animals, isn't it? So now, if you happen to understand the structure of the DNA, so DNA is made up of double helical structure. So you can see this is the structure of the DNA. So usually DNA is made up of some chemical bases. So those chemical bases are generally, they are nothing but adenine, thymine, guanine, and cytosine. They are complementary base pairs, we will call. So always adenine will be accompanied with thymine, guanine will be accompanied with cytosine, like that. So they are nothing but the complementary base pairs, we will say. So if there is A here on other strand, so then it would be accompanied by T. So like that we can have the complementary bases. So that is one thing. So this is how the structure of the DNA will be there. And this particular DNA, if you see, it is made up of genetic information in the form of codons. Let's say, for example, AGA or AGC, so it would be in the form of triplets. So it is nothing but codon or genetic information in the form of pockets, so codons. So if we happen to see on the chromosomes a particular gene sequence, a particular genetic information or a particular DNA sequence can be exactly located on the specific location of the chromosome. So such kind of exact location where we can see a set of DNA sequence or genetic sequence, basically like the flags that you can see here. So in this particular chromosome, if you can check and if you can go there and if you can see, you can find out the gene responsible for that particular character. Let's say, for example, gene responsible for the ABO blood group, or gene responsible for the RH factor. So all of those things, if you happen to understand or if you happen to see, they can be located or they can be seen on the exact location on a chromosome. So now, what are genetic markers means? A genetic marker is nothing but a gene sequence or a DNA sequence with a known location or with a specific location on the chromosome. So that's what the basic meaning of the word genetic marker. So here what you can do, what you can find, so on a chromosome at the exact location you can mark the location of a particular gene. And again, what is the function of a gene? Gene will be expressed in the form of a protein. So each gene translates into a protein. So that's where each gene is responsible for one function. So one gene, one function. So for every gene, one function will be there. So that's how, if you want to find out the a gene with a particular function, then genetic markers, particularly we can just locate them on a particular chromosome in a systematic fashion. So they are nothing but genetic markers. So what they do, so you can see here, for example, if this is the if this is the DNA structure, so on this particular spot, you can locate the genetic marker or you can locate a particular gene. So these are nothing but genetic markers. So what do? They act as landmark or sign spots on the genome. So what do you mean by genome again? The complete set of genes of a particular organism is known as genome. So within the genome, so genetic markers will act as landmarks or sign spots. So as they are helping as the genetic landmarks to identify something, so we can track the differences in our genetic information. So that's how they'll systematically highlight the variations among the populations. So that is another important information. So what do they do? So genetic markers are generally useful in identification of the genetic variations among the populations. So how, why? Because genetic markers are highly polymorphic in nature. What do you mean by polymorphic? Poly means many, morphs means forms. So they are present in multiple forms. So that means they show variations within themselves. So let's say, for example, so ABO blood group. So a person can have A blood group, a person can have B blood group, a person can have O blood group, a person can have AB blood group. So blood groups are, although they are different, but here they exist in multiple forms. So they are not in single form like that. So such existence in multiple forms are showing variations are nothing but polymorphic expression. So genetic markers are highly polymorphic. Therefore, if we systematically study genetic markers, then we can systematically identify the genetic variations. So I hope you are able to understand the concept. Now, what can we do? So genetic markers are nothing but they are the powerful tools in understanding human biological diversity and in understanding the forensic analysis and also for studying the disease association, etc. So with the help of genetic markers, we can do all of those things. So that's where the question has been given and picked. So now let us try to understand further aspects about the genetic markers. So if we happen to understand example, so what are the popular genetic markers that we can have? So popular genetic markers that we can have is that one are nothing but allozymes. Allozymes are nothing but different forms of the alleles of the enzymes. So allozymes. Then we have studied about something mitochondrial DNA, the DNA which is present on the mitochondria of the cell. So that is known as mitochondrial DNA, right? And then microsatellites, mini satellites. So they are all nothing but the small tandem repeats of the DNA sequences. So that's one thing. Then restriction fragment length polymorphs. So RFLP we will say, restriction fragment length polymorphs. So a fragment of the DNA sequence or nucleotide sequence will be repeated. So that is nothing but, that will be in variation. That is nothing but RFLP. Then random amplification of polymorphic DNA. So we'll call it as RAPD. So random amplification of polymorphic DNA. So different forms of DNA. Then amplified fragment length polymorphism. So polymorphs. So they are all generally examples of our genetic markers. So in common terms to say, these type of genetic markers, they will be responsible for certain aspects. Like we can call ABO blood group as a genetic marker. RH factor, RH blood group, blood system is also a genetic marker. HP, heparin is a genetic marker. HLA, so that is another genetic marker because they exist in polymorphs. So they are all examples, right? So let's try to just know some of the genetic markers. So first, if you happen to understand what is single nucleotide polymorphs, single nucleotide polymorph, nucleotide polymorph. So what happens here? So there would be variations involving a single base pair in the DNA sequence. So single base pair would be experiencing differences. So that is single nucleotide polymorphs. Then short tandem repeats. So this would be another type of genetic marker that we can have. Now, what are these STRs? So they are also generally called as microsatellites. So this was also one microsatellite, mini satellite, such kind of questions also been reflected in prelims also. So try to recollect such aspects. Now, this short tandem repeats are also called as microsatellites, and they are short. They are the repeated sequences of 2 to 6 base pairs. So short repeats will be having, we can see in the DNA sequence. So there, they'll be repeated at regular intervals. But what is the key here? So they'll be having some variations. So therefore, we can call microsatellites also as genetic markers. So that's one important information. Then variable number tandem repeats. So this is another important genetic marker. So VNTRs we'll call. So similar to these short tandem repeats, VNTR are also same, but thing is that they'll be longer repeated sequences, nearly 10 to 100 base pairs will be repeated. So this is the basic difference between short tandem repeats and variable number tandem repeats. So in short tandem repeats, you can have repetition of 2 to 6 base pairs and with variations. In variable number tandem repeats, what you can see, you can have multiple variations with respect to the, there would be like repetition of 10 to 100 base pairs in the DNA structure. So that would be the examples for the genetic markers. So try to remember these things. So common examples we have understood. So blood systems are like HLA system or HP, heparin. So all such things are examples. So you can see the basic classification of the genetic markers. So genetic markers can be basically classified into different types. One is like the chromosomal markers or lineage markers. So where we can see certain genetic markers on the X chromosomes and certain genetic markers on the Y chromosomes. the genetic markers that are there on the X chromosome, you can see particularly with respect to the example is that mtDNA, mitochondrial DNA. So with the help of mitochondrial DNA, we can systematically assess and we can systematically track the maternal lineage, because mitochondrial DNA will be inherited from the mothers only. So that's where it helps to track the maternal lineage. Hope it is clear to you. Now, if you see, there are some Y chromosome markers. So specific genes will be inherited from the father. So those Y chromosome markers will help us for understanding the paternal lineage and to understand the paternal ancestry. So that's one thing, right? So they are nothing but chromosomal markers or lineage markers. So they are one set of genetic markers. Is that it clear? Now, there are certain genetic markers which are called as repetitive markers. Repetitive markers. So what are these repetitive markers? Short tandem repeats. We have understood short tandem repeats. So short tandem repeats are also called as microsats or microsatellites. So these are one type of repetitive markers. And many satellites, so which are called as the long repeats. Let's say, for example, variable number tandem repeats. So they are long. So 10 to 100 base pairs will be repeated and they will show variation. So do you remember that? So mini satellites or variable number tandem repeats. So that's another important aspect, right? So you have to remember this thing also. Then if you happen to see, so we can have DNA sequence variants. So DNA sequence variants. So what is this? So we can have this small nucleotide polymorphs are like restricted fragment length polymorphs. So these are DNA sequence variants, variations with respect to DNA sequence. So that is one thing. So these are nothing but some of the examples of the this is how we can classify the genetic markers. So try to note it down somewhere and try to have it in your notes. So this is one thing. This is how, so we should understand the genetic markers. So genetic markers are all about this. So so that you can remember thing. One thing is that what is the genetic marker? Now you just look. Now how you can understand this in a very simple term means try to just understand this. Now, if I give you a marker and I ask you to locate the map of India on world map, so then you will pick the marker and you will just place at a particular point at location and you say that this is where India is located. Can you say that? Why can we say that? Because India has a specific location on the world map. So in the similar fashion, on the chromosomes also, you can just locate certain genes with exact location. So those gene sequence or those DNA sequence with known location or known loci on the chromosomes refers to genetic markers. So that's it. So you have to remember it. So now let us proceed further towards the applications of the genetic markers, right? So what are the applications? So we have understood that genetic markers will be helpful in understanding the population variations. So the population variations that are existing among the different people or different human beings, that itself is known as genetic markers. Because friends, genetic markers are known for highly polymorphic characters. That means they show variations in the human beings. So they represent variation. So that's how we can understand the population variation systematically with the help of genetic markers, right? So yes, so we are getting a question. So I'll just address the question at the end, right? So then what are the population variations? So how it will represent? So genetic markers are fundamental tools for studying population genetics and human evolution and population variation. How we can help, how genetic markers help in that process? Let's see. So first and foremost thing, genetic markers will help in reconstructing the migration patterns and migration routes in the population, right? Then, how we can just understand the migration pattern or migration routes? So there, when we try to systematically study the Y chromosomal inheritance and X chromosomal inheritance linked DNA. Let's say, for example, we have mitochondrial DNA or we have Y chromosomal DNA, so which are peculiarly inherited from either father or mother. So if we systematically study the mitochondrial DNA and its inheritance pattern and its, if we can track that, so we can systematically assess the migration routes. So that's where you can see, so uniparental markers like mitochondrial DNA or these Y chromosomal lines, so they are studied to assess the maternal or paternal lineages. So while the autosomal markers will help to understand the complete ancestry. So that's how we can study the ancestral pattern, and this will further help us to understand the migration patterns of the population. That's one thing. Then the genetic markers also will be helpful in understanding the adaptations of specific populations. That's very, very important aspect. So now how they will help, look here, so certain markers are linked to high altitude intolerance, tolerance, and lactose persistence or pathogen resistance, etc. So what do? They help us to explain about the population specific adaptations. So specific DNA, there is like permanent adaptations. So acclimatization and adaptation. Acclimatizations are nothing but temporary changes that we can have, temporary modifications of the animals to the environment. Adaptations are not like that. Adaptations are permanent and long-term changes. So from genetically also there would be changes. So in such a fashion, certain genetic markers are linked with certain climatic or environmental adaptations. So with that, we can also understand how adaptations will be there. So that's one another advantage or application in understanding the variation. Then measuring the genetic diversity. So genetic markers also helps us to understand the genetic diversity. What do you mean by diversity variations? Genetic diversity means variations among the genetic composition and genetic aspects of the animals or humans. So here genetic markers will be employed generally in genealogical DNA testing. With the help of the genealogical testing and analysis, what we can do, they determine the genetic variations between individuals or populations. So that's how we can systematically understand variations among the populations, because they are known for polymorphic nature. So existing variations, they shows variations. So that's why populations also show variations. So what are genes basically? They are the information carriers. They are inherited from one generation to another generation, isn't it? So these genes will be represented in the form of protein. So they will be in fact represented or manifested in the various activities. So that's how population variations do exist, and we can study them with the help of genetic markers. Then what about the role of genetic markers in understanding the disease association? How we can just predispose diseases? So this is another part of the question. So how you can address and answer this? So we can say that in medicine, generally to say, genetic markers are crucial for identifying genetic basis of inheritance and complex diseases. There are certain complex diseases and there are certain genetic diseases. So with the help of genetic markers, we can systematically identify such kind of diseases. So first and foremost, if you see, so genetic markers will help in the concept of gene mapping. So what is gene mapping? So when we systematically map all the genes which are located on the chromosome. So that is nothing but gene mapping. So what do we do here? So markers are used in linkage studies and, look this statement very clearly, genome-wide association studies. So genetic markers will help in linkage study, X-linked studies, Y-linked studies, like that. So genome-wide association studies. So this is in fact another important question that has been asked in same 2025 itself. So the paper has some lot of connectivity. In 10 markers, you can see mitochondrial DNA question. In 15 marker, in 20 marker, you can see this question that we are discussing, genetic markers. And again, you have this question, genome-wide association studies. So we'll be getting that in the next, in later days. So this is one thing. So we will be studying this in detail. Now, the markers are used in lineage studies and genome association studies. What is the chief purpose? To find associations between specific marker and disease. So we can find the association, how a particular gene will be responsible for a particular disease. So that's how one important function of it. Then with the help of genetic markers, we can also identify the predisposition of the diseases. Predisposition of the diseases means before the hand we can just understand the diseases. So what are they? Like, let's say for example, some of the complex conditions, like let's say diabetes or heart diseases or certain cancers. So we can systematically identify them by systematically studying genetic markers. So if we can identify genes that are responsible for occurrence of certain cancers, if we can identify genes that are responsible for occurrence of diabetes, if we can identify genes that are responsible for occurrence of certain heart diseases, so we can identify and we can know them. So that's how it is, we can predispose these diseases. That's one thing, right? Then further thing, we can also have the personalized and predictive medicine with the help of genetic markers. So based upon the patient's unique genetic profile, so what we can do based on this unique profile, so we can, doctors can enable and doctors can select the most effective treatment modality based on the unique genetic profile of that particular person. So certain drugs will not work for a particular patient. Only certain drugs will effectively function for certain patients. So if you systematically study genetic markers, they further help in such kind of personalized medicine making it or predictive medicine. So that is one thing. So this is how genetic markers will help in the disease association aspect, right? And then further, if you happen to understand the role of genetic markers in forensic analysis or forensic science, so what we can have, so particularly if you see things like short tandem repeats, STRs, so they are the cornerstone of modern forensic science. Why we are calling? Because we can use the genetic markers in multiple areas to understand. So let's say for example, we can use the genetic markers in DNA fingerprinting or profiling. So for identification of the suspects. So we can match the sample with the DNA profiling exactly, and we can identify the criminals. So for criminal identification, we can use this. So genetic markers also will be helpful in this, in that aspect, because they are located exactly on the same chromosome. So we can pick the sample and we can see that on 19th chromosome, so at this particular location, this gene will be there. So if you look, if it is getting matched, so exactly, that's how we can catch the criminals also. So that is done. So to say here, for accurate matching of the biological samples from the crime scenes and human identification, we can use genetic markers. So in DNA profiling and in DNA fingerprinting, we can use it. Then for disaster victim identification, so aftermath of occurrence of disasters, let's say recently there was a bomb blast. So there was a bomb blast in the Delhi. So it created ruckus, and which has led to loss of more than at least 10 to 15 persons. So in all such kind of disastrous situations, so what we can feel is that we can identify the victims with the help of these genetic markers. So that's how we can recognize the bodies which are unknown. So that's one thing where like genetic markers will be helpful. Then paternity dispute resolution. So one thing like paternity disputes, could be arise in the main course of time. So to solve such kind of issues of paternity disputes, so we can also use the genetic markers. And in other ways, like the personal identification, so to solve the medico-legal cases. So that is another important aspect. So in all these aspects, our genetic markers will be helpful and they'll be useful for us. So that's with respect to the applications of the genetic markers, right? So now the Pragnya Isha is asking, so can come again please explain types of genetic markers. So if you happen to see the genetic markers, so genetic markers types of genetic markers, right? Okay. So genetic markers can be systematically classified into 3 types. So one is nothing but chromosomal markers, other one is nothing but repetitive markers, and the next one is nothing but DNA sequence variants. So what are these chromosomal markers? Chromosomal markers are also known as the lineage markers. Those genetic markers which can be present in the X chromosomes only and which can be inherited from the mother only. Let's say for example, the mtDNA, mitochondrial DNA. Mitochondrial DNA is presented exclusively on the mitochondria of the mother. So that's where with the help of this one, we can trace the maternal lineage. So that's why we are calling it as chromosomal or lineage marker. Then certain markers will be there, genetic markers will be specifically seen on the Y chromosome. So they are known as Y chromosome markers, right? So that is one thing. So these Y chromosome markers will help to trace the paternal lineage of the persons. So that's why we are also calling them as lineage markers. Then there would be certain genetic markers which are known as repetitive markers. What are repetitive markers? So short tandem repeats or long tandem repeats. So you look here, short tandem repeats means what? Certain DNA sequences, let's say for example, ACC, TTT. So we can have such things. So where you can have things like, let's say for example, ACC, ACC, ACC. So this is a kind of repetition. It's a repeat. So what happens in short tandem repeats? Simply 2 to 6 pairs of DNA will be repeated like that. Short tandem repeats. That means randomly repetition would be there. And long tandem repeats means like this ACC, 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 at multiple places you can see nearly 10 to 100 times repetition. ACC, ACC, ACC like that 50 times. In another spot you can see ACC, ACC, ACC like that 50 times. So that's how they are known as the long repeats. So this is how they will be repetitively in nature. So that's why we will call them as repetitive markers. Then DNA sequence variants. Sequence variations will be there. So sharp, small nucleotide polymorphs. So they are nothing but like within a base there would be changes. And restricted fragment length polymorphs. A particular length of the DNA fragment will be repeated. You can see this single polynucleotide polymorph, that is variations involving single base pair in the DNA sequence, and restricted fragment length polymorph. So what is this? Shows a fragment of the DNA will be showing variations and it will be represented in different forms. So that's how this will be shown. So these are nothing but different types of genetic markers. So I hope it's clear to you. So that's with respect to the genetic markers and types of genetic markers, right? So friends, let's try to move towards the conclusion. So how we can conclude the answer? So with some futuristic or with some sort of the current relevance or the further scope and significance of genetic markers, you can conclude the topic. So how you can do that? So let's say, for example, genetic markers are seen as essential tools for studying human diversity, for identifying disease-linked genes, and enabling precise forensic identification. So they are all nothing but some of the prospective examples, so prospective applications. So we can quote them again. So we can reiterate the significance of the genetic markers. And then they bridge anthropology, medicine, and law enforcement by revealing our evolutionary history and health risks and individual identity. So you can use such kind of statements where we are showing the scope of the genetic markers. The scope of the genetic markers is not only restricted to the medicine field or law enforcement field. So in anthropology also, we will help it in the evolutionary history. So that's very important aspect. In anthropology, we can see evolutionary history. In medicine, we can see health risks. In law enforcement, we can see individual identity. So this is how you can break or you can bring in the scope of the genetic markers application here again. Then finally, what you can do with advancing technology, their accuracy and lawfulness continue to grow, making them indispensable to the modern science and society, particularly the forensic science. So that's how you can say that the genetic markers are helpful not only in the forensic medicine, forensic science, they also help in the genetic disease disposition, and they also help in understanding the population variations. So that's all, and that's how you can conclude the concept. So remember these examples. ABO blood groups is an example for genetic markers. RH blood groups is an example for genetic markers. And HLA system is another example. So all of these things are mentioned in your syllabus also. Try to just study them in a systematic fashion, right? So that's all for the day from my side. If there is any question or if there are any further doubts, you can just go on and you can just text in the chat box. Is that it clear, or shall we wind up the discussion? So within the time, let's see, like I insist again, like some of you have started writing and some of you are still yet to start write the answers. So those who are about to write, do it immediately, because sometimes later becomes never. Remember this, right? So some of the students have started writing and have started receiving their copies also. So for all those who have started, and I can see some of the improvements in your writing also. So keep writing and keep learning. So we'll meet again. I hope like there are no further doubts. So let's meet again tomorrow at 11:30 a.m. So if you want to just download and get more such information, you can join our Telegram channel, Anthropology Guide, either through this link or through this QR code. So that's all with respect to today's discussion. So friends, we'll meet tomorrow exactly on 11:30 a.m. and let's discuss another important question, and let's try to decode every question and let's master every concept. Thank you all for joining with me for this session.